Hi guys, I'm Chloe.
And I'm Josh.
And today we'll be talking to you about silica column purification. Silica gel is used in column chromatography because it can adsorb basic substances that are within a material that have to be purified or separated. So what is it used for? Purification of genomic DNA, amino acids, lipids, and steroids. Spin column cased nucleic acid purification, also DNA extraction procedures can also be performed with this technique. Gel extraction kits use silica columns, as well as PCR, which is also polymerase chain reaction purification. And then silica gel is very easily available commercially in different sizes and forms, and it can be regenerated or reused numerous times, therefore lowering the costs of purification significantly. So what is the underlying science behind silica column purification? So the basic principle of column chromatography is just to adsorb solutes of a solution with the help of the silica, and then further separate the mixture into distinct components. So silica gel is a polar adsorbent, which means it adsorbs other polar materials. So it has a high specific surface area, and it is the stationary phase for column chromatography separations. So just a side note, hydrated silica gel surface can have three types of, it's called silanol groups. They are free silanol, geminal, and associated silanols. And these have highly acidic natures, and so that increases the retention for basic compounds. And so during the column chromatography, the compound that does not travel the fastest in the column means that it is adsorbed more strongly to the silica gel, and that polar compound is what it is. And so the greater attraction between polar solvent and polar molecules is present. And lastly, silica remains neutral and doesn't interact with any of the substances that pass through it. So now Josh is going to take you through a general procedure for silica column purification.
Okay, to start, first we're going to need a reaction column, stopcock, a beaker, there's a couple beakers on hand, and then a ring stand. You're going to attach the stopcock onto the reaction column and then support that by the ring stand, put a beaker underneath. And then afterwards, you're going to put a little bit of cotton inside and make sure it, just not too much, but enough so it like kind of covers and fits snugly inside the little curves inside the reaction column. After you put the cotton, add about 1 centimeter layer of sand on top of it. Then off to the side, you're going to obtain a certain amount of silica. Make sure you do this whole thing under the hood, so that way, because there's a severe health hazards when using silica with breathing. So once you obtain your required amount of silica after you recorded the mass, you're going to put that into a beaker. Then you're going to add some column solvent into that. You're going to mix it up with the stirring rod, so you should get kind of like a slurry kind of consistency. Once you have that, you're going to put a funnel on top and pour the slurry down into the reaction column. And then once you see that, you should see kind of 2 layers forming, the silica and then a solvent layer, because after all, this is like a slushy kind of thing, so it's not perfectly mixed. So once you see that, you're going to start turning on the stopcock and then drain out the solvent, which is like the liquid above it, until it's like a little bit above the silica layer, just a little layer. And then once that little solvent layer is there, you're going to add another layer of sand. It's like about 1 to 2 centimeters thick. You're going to put that on top of the silica, and you should still see that little sliver of the solvent layer on top. Once that is in there, you're ready to put your sample in. You can use the glass pipette and pour your required amount of sample onto the reaction column. Once that's going, you turn on the stopcock and start draining the everything, all the solutions, and so you should see the samples dripping down. And then you should see just a little bit of the sample and solvent intermingling and going into the sand. You should see a little bit. After that, add a little bit of the column solvent, and then you let it go through. Let it go through again, and then after you put a little bit of the column solvent, you should see a new layer forming. So you got the cotton, then you got the bottom layer of sand, the silica, and then you should see another new kind of layer formed between the silica and the top layer of sand, then the sand, and then you have your solvent layer, which you're gonna add a bunch more into that reaction column. After you add that, you're gonna turn on the stopcock again, and then you're gonna allow the whole like solution to drain out into the test tubes. You're gonna collect them and make sure that the test tubes are about half full. If it's over that, then you just get another one. So you're gonna let it go for a while until all the solvent layers gone, and then everything is collected in the test tubes. Next, you perform thin layer chromatography on all the test tube solutions, and then you're gonna analyze your results, and whichever one has like one spot on them, those are the ones you're going to use. And you're gonna combine the solutions that have the one spot into one giant little test tube, and you're gonna put that into a rotary evaporator, so that way it can separate the solvent, and then afterwards you get the products.
That's it for now. So thanks for watching our video.
Bye.